All right, so as we said previously, endocrine system, we'll do a quick recap of the stuff before spring break and then learn some new things. Before spring break, we were looking at the fact that we have a weight problem in the US. Keeps getting worse. You might remember we made even new colors along the way as the years went on, as we got more and more obese. And from there, we started trying to figure out, is there a metabolic reason for that? Other than just like we don't exercise enough, we eat too much, that type of thing. Is there something that drives that? And so we saw some of these mice that were very overweight, like this guy. And we can see this one next to a normal mouse as a point of comparison. Saw this one. It's actually a true picture also. And so then what we did in these experiments is we were trying to figure out what was causing these mice to be like that. And so a few things that we knew was up here in the brain. So let's say this is the brain. Specifically the hippocampus. The hypothalamus. Hippocampus is memory. So on the hypothalamus, we have receptors. And those receptors are for a chemical called leptin. So I'll just call these LR for leptin receptors. And what happens is I have fat cells in my body. So there are some fat cells. And right now they're not very full. That's why they look kind of like deflated balloons. But whenever I eat, then my body is going to burn some calories right away. If I have sugar in my bloodstream, then I could use that now. And if I have extra sugar, then I can store it as this substance, this kind of oily substance called glycogen that I store in my liver. And then eventually, if I want to store some calories more long-term, we know that lipids are kind of the more long-term solution. So I'll start filling up these cells with lipids, with fat. And so as they do that, they kind of plump up, of course. So now my cells look more round because it's sort of like a water balloon, except it's more like a fat balloon, and it fills up with fat, which is disgusting. And so I fill that up with fat. And then when I do that, my body releases, or my fat cells release this hormone called leptin. And one thing that'll be weird about this, since I don't get to like back and forth with you guys, is sometimes I'll ask questions, pause for a second, that way if you're at home you can try to see if you know the answer, and then I'll give you the answer. So it'll be super awkward. You think it's awkward for you, I'm talking to a phone. So we release leptin, and what do we know about leptin since it ends in IN? Yes, that's exactly right, it's a protein. Things that end in IN and EN are typically proteins like insulin, leptin, actin, myosin. And so what will happen is my fat cells, as they get full, they release leptin. Leptin is a chemical released into the bloodstream. It'll eventually go and hit that leptin receptor. And when it fills that leptin receptor, remember it's a receptor, so that receptor has a specific shape for leptin. So it has to chemically and physically fit. It physically fits a little bit like a lock and a key, but it's not stagnant. It's not as rigid as a lock and a key. So most books will refer to it as an induced fit. So I might have a receptor that, fits like this, and then whenever leptin goes in there, that receptor might change its shape a little bit, for example. And so what will happen then is my brain then creates a feeling of, I feel full. Okay, well, let's say that I just have one fat cell that filled up versus 2 versus 3 versus 4. So if I start filling up more and more and more fat cells, that guy and that guy and that girl, I'm not sexist against fat cells, then the more fat cells I fill up, the more leptin I release, the more of a feeling of full I'm supposed to get. Now, just to be clear, this isn't the only way that we feel full. We also feel full when our stomachs are just full of something. That's why if you're really hungry and maybe you're also thirsty, and let's say that you drink 20 ounces of water for just a little bit, you'll feel full. You might even drink so much liquid that you're like, oh, I don't feel hungry anymore. Pretty soon your body realizes that you didn't get any calories out of that water, but you'll feel full. Blood sugar can also, when it's high or low, I can feel full or I can feel hungry. So this isn't the only thing that tells us we feel full. It's just a thing that tells us we feel full. And so what we were doing with these mice is they were trying to figure out, this bigger mouse over here, is his problem that he has faulty leptin receptors, or is it that he has faulty leptin, the chemical? So does he have a mutation in the gene for leptin maybe, or a mutation in the gene leptin receptor? So what they did was, again, we saw this in class, it was kind of a troublesome experiment, but we do experiments on, if we consider humans higher than other animals, then to us it's worth it to do experiments on mice if it helps humans. So what they did is they had these two genes. One was called OB and one was called DB. OB is linked to obesity. DB is linked to, no, that's inappropriate, diabetes. And so what they were trying to figure out was they basically had it isolated to where they're like, all right, we think that one of these is coding for the receptor and we think one is coding for leptin, the actual hormone, the protein hormone. So they did this experiment with parabiotic mice, and in parabiotic mice, what they do is we basically kind of do a surgery. I say we, I didn't. But they sew these mice together. So picture conjoined twins. Well, now you're just making conjoined twins out of mice. So if I'm a mouse and this person's a mouse, you kind of cut my side open, cut their side open, and we start intertangling their systems. So our bloodstreams, we're sharing the same bloodstream. And then you kind of sew us together enough to where if I kind of turn left and my buddy turns right, that we don't rip apart. And so that's what they look like. So here's some parabiotic mice pictures over here on the left and on the right. Here it's a little bit easier to see because they have different fur. So they kind of sew them together, and then later on the more light colored one was removed. The next picture is a little bit rough if you don't want to look at it. They're sewn together. Here you can see before they put the skin back on. And so what happened when they did this was what you can see in this diagram. So I got the big guy and the normal guy. When I sew them together, the big guy shrinks down. Over here I've got a big guy and a normal guy. He has a DB mutation. This guy has the OB mutation. So that means whatever the OB gene codes for, this guy has a mutated version. Obviously it's mutated because he's not supposed to be that big. This guy has the DB mutation, and whenever you sew him to make a parabiotic mouse with a normal guy, then the normal guy gets even smaller. Over here, when I sew an OB mutant and a DB mutant together to make parabiotic mice, the OB guy gets smaller. So based on that, we are trying to figure out what codes for the receptor, what codes for the actual protein, OB or DB. So pause the video for a second once I ask it, because I'll answer it right away. But if I'm looking at this guy based off of this, who's the mutant leptin protein? Is it OB or DB? Who's the mutant leptin receptor? Is it OB or DB? So give it a shot. Pause. All right, and we're back. So what we find is this. Because this guy shrank down, that means that his receptors are working okay. So if you think about it, if OB means that I code for leptin protein, and DB codes for leptin receptor, then that would explain these pictures. So this guy has normal receptors and normal leptin. If this guy just doesn't make leptin, then when they're sharing the same bloodstream, they're sharing leptin protein. They aren't sharing leptin receptors, but they're sharing leptin protein. So now leptin from this guy is hitting his normal receptors and he's shrinking down. So that means that his problem is he doesn't make quality leptin. What's the cause? Who knows? Some sort of genetic mutation that means that he doesn't make quality leptin. But the exact mutation, I'm not sure. Same thing over here with DB. If he has messed up receptors, it doesn't matter how much leptin he gets. He's going to stay the same size. What's interesting is this guy got smaller. So your body's constantly, or his body, is constantly trying to maintain homeostasis. So essentially what will happen is if you are sending out leptin and it's not hitting the receptors, we get caught in this feedback loop. We'll learn a little bit more about failures in feedback loops with iodine and some thyroid hormone later. But what will happen is now his body, because he's not hitting the receptor, he starts secreting more and more actual leptin. So he has extra leptin circulating in his bloodstream, and that's why this guy gets even smaller, actually to the point of death if they let the experiment go long enough. And back over here, similar idea, because OB has a leptin chemical deficiency, then when I hook him up with this guy, he'll shrink back down. So we talked about that right before break, and then from there, we went into Zara. Zara, we got to in some of the classes, not all the classes. Zara, if you want to look up online Zara lipodystrophy, you can, L-I-P-O, like lipid, L-I-P-O, dystrophy, dys, and then trophy. So Lipodystrophy, lipodystrophy. So Zara in that picture is 13 years old. She looks a lot older. And again, if you Google Zara lipodystrophy, all the hits that come up are articles or videos on her. We had a video clip that we showed in class that was a few minutes long, if you'd like to view that. But the problem with her is that her body is not storing fat in her face, and so her face looks kind of gaunt. That's why her skin looks really, really thin there. It's because of lipodystrophy. She's not properly storing fat in her face. The next thing that we looked at was actually a review from earlier in the year also, was 2 major ways that hormones can work. So we kind of had these 2 different versions of pictures that looked like this. You've also got these in your notes, slightly different versions, but right there in your notes. And what was it that was different between these 2? That's exactly right. It turns out that the location of the receptor is the difference. So here, my receptor is actually buried in the surface of the cell. It's a membrane-bound receptor. Here, my receptor is inside the cell. And so what do we know about that particular hormone for sure if the receptor is internal into the cell? By the way, sometimes these receptors are actually in the nucleus to begin with. So what do we know about that specifically? Well, these guys have to be small enough and non-polar enough to actually get through the cell membrane. Remember, small things get through the cell membrane easier than large things. That makes sense, squeezing through the phospholipid bilayer. And then the more non-polar you are, the easier you can get through the cell membrane. So the bigger and more polar you are, the less likely you are to get through. So maybe this signal molecule, this hormone, is really, really polar. Or let's say that it was super large. It can't get through the membrane very easily. So it has to hit a receptor on the outside. So you'll notice the difference is this guy squeezes through the cell membrane, hits a receptor, and then goes in and typically these guys will go in and turn a gene on or off. So they might act as a transcription factor. They might inhibit a gene in some way, and we can turn a gene on or turn a gene off. That's what they typically do. That's not the only thing that they have to do, but that's what they typically do. The majority of cell communication happens this way, though. Not this way. This way. Most of the time, cell communication has a polar molecule, and it hits a receptor that's in the membrane. So they'll call it a membrane-bound receptor. They might call it an external receptor or extracellular because it's facing the outside. Intracellular, I-N-T-R-A. Intra means within, kind of like an internet is between networks. The intranet is within. So in Keller ISD, we have an intranet, and I can only access that when I'm actually on school property. Internet, that's between networks. So I can, get on the internetwork and, order something off of Amazon or whatever. So another big difference is this. Both of them have receptors. Again, if you're talking about reception, the receptor and the ligand, ligands are what fit receptors, they have to fit together chemically and physically. Another major difference, though, is that this one right here has a transduction pathway, typically. So I'll have a membrane-bound receptor, and it'll activate a protein, which activates a different protein, which activates a different protein, which eventually does something. So in my notes, I kind of break it down into 4 steps instead of 3. I say you send out a signal molecule, probably a hormone, in this chapter anyway. Then there's reception, then transduction, and then a response. If you're writing on the AP Bio test, by the way, they love comparing and contrasting these two on the AP Bio test. If you're writing about these on the AP Bio test, again, reception has to be specific for its ligand. Transduction is a protein activating a protein activating a protein. It might activate second messengers. Those are soluble small molecules that are not proteins, like calcium, cyclic AMP, CAMP. And then in the end, a response. And don't just say like there's a response, give an example of a response. If I'm now turning on a gene, if this is growth hormone or something like that, then that would turn on a gene that says, grow. I might turn genes off as well. Okay, a couple other things. This guy right here that's lipid soluble, we call them steroid proteins because steroid proteins typically come from cholesterol. And if we look at the structure of cholesterol there, we'll notice these steroid proteins like cortisol, aldosterone, testosterone, estradiol, progesterone, they all basically are modified cholesterols. We saw that. Oh, one last thing on this. Not only do we do transduction, but what else did we do when we turned on a protein that turned on another protein that turned on another? We did amplification. So really, one protein might turn on many, and each of those turns on many, and each of those turns on many. So if this turns on 10, and each of those turns on 10, and each of those turns on 10, that's 10 messages times 10 more is 100, times 10 more is 1,000, and so forth. Why have all these steps in between? Each step is kind of a checkpoint. So if the final action of this was to have me, the organism, grow, and I want these cells to multiply, to do cell division, then I might want to make sure, do we have enough of this nutrient? Do we have enough of that nutrient? Do we have enough of this nutrient? And if the answer is yes, 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 then we go. If the answer was no on any of those, that's kind of saying a pause. That's like, don't grow yet. The example that I've given in class is when it's time to make the next iPhone, there's somebody working on the glass, somebody working on the battery, somebody working on the processor, whatever. And does the glass scratch as easy as it used to? No. Okay, cool. That's the answer I was looking for. Does the battery last longer? Yes. Okay, cool. That's the answer I was looking for. And I check with all my engineers. These are my engineers. And if I get the right combination of yeses and nos, then we produce the iPhone. If along the way somebody's like, no, the battery explodes, then what I should do is I should say, all right, he's saying pause, so I don't do the final response of producing the phones. Okay, one last thing that we got to in most of the classes, and then we'll be on to new stuff. New stuff is always the most fun stuff. Was this. If you flip to the next page in your notes, up the very top, the first hormone we're going to look at is growth hormone. On the AP Bio test, supposedly you're not going to have to know any specific hormones. It wouldn't surprise me at all if they talked about hormones, but gave you enough information in the actual question stem to answer it. The most common hormones that they would talk about would be insulin and glucagon. They do opposite things. We'll learn about those. But we're going to kind of hit some of the major hormones that you might come across in a biology class or an anatomy and physiology class. So again, we'll start up there at the top with growth hormone. So this is Robert Wadlow. He died right around 1940, and he was born right around 1920. He died at age 22. And right here, Robert is with his dad and some other dude. I don't know how old the dad is in this picture, and I don't know how tall he is. Sources that I find always just refer to him as an average-sized man. But Robert Wadlow right there is age 12, and he's 6'11". Most doorways in your house are 7 feet. At Timber Creek, our doorways are 7 feet. So, you know, factor in an inch of hair and he'd be brushing the doorway when he's 12, you know, gonna be heading to high school in a few years. Here he is again, he's gotten, you know, taller. There's Robert, there's his dad. Kind of impressive. Here he is. I'll put a, I'm going to have a page with video links that will be in the files on our Canvas page if you want some of these, but I'll have a link to, it's about a 3-minute video of Robert Wadlow, and I'm going to show you a little bit of a video clip on Robert Wadlow also. Here he is in a class photo. He's the guy in the back. There he is bumping into a chandelier with his chest, and that's probably some relative. If not, that picture makes me, I don't know. It's creepy. The next one is Robert with some of the ladies. Here he is again. Here he is at the elevator. This one I don't know how well you can see, but they're unloading his casket off of a flatbed truck, and it's large. And then here's a growth chart. So since I'm new to this method, I'm not sure how well you can see it, so I'll read some of them to you. When he was born, he was 8.5 pounds. That's pretty typical. I don't know what exact average is, but average is probably in the US somewhere between 7.5 and 8 pounds, probably closer to 7.5. I weighed over 8 pounds when I was born. At age 8, he was 6 feet tall. So I'm 5'10 and a half, so I say 5'11". So he was taller than me when he was 8. When he was 12, he was 6'11". When he was 14, typically you enter high school when you're 14, he was 7'5" and weighed 300 pounds. When he was age 21, he was 8'8" and 492 pounds. When he was 22 and he died, he was 8'11" and he was 439. So the thing there's a lot of interesting things, but one of the interesting things is when he was 8'8" and weighed 492, he always looked skinny. Like every picture you see of him, he never looks thick. You know, like he looks very, very skinny no matter which picture you look at. And that kind of lets you know just how tall he was, because when he's 8'8" and he's almost 500 pounds, like he's a skinny looking dude. So pretty impressive. And so the cause for this, I think we only got to this in 1 or 2 classes, is the cause of this is on your long bones, like this right here is a shin bone, your tibia, and that right there is a femur, a thigh bone. And up at kind of the tops of them, you can see, in slang we call them growth plates, but at the tops and the bottoms, you can see these growth plates, the epiphyseal plates. That's where your body adds new bone. And so my son is 12 and he's taller now than he was when he was 10. And so when his long bones grow, you don't add bone just anywhere, you add it at the epiphyseal plates. So for this shin bone to get longer, we can add cells there and we can add cells there. That's where they grow. When you're 15, these growth plates are open. On an x-ray, there's more cartilage in there and less bone, so it actually looks like there's kind of a dark spot. Like the bone is white, of course, and cartilage is more grayish, and there'll be kind of a grayish dark line going through there because it's not closed. It's not a closed growth plate. So that's why for those of y'all that maybe when you were young, you broke your wrist or your elbow or something like that, or an ankle, and they look at an x-ray and they're like, oh wait, is it on the growth plate or not? If it's not on the growth plate, if it breaks right there, cool. Your bone breaks, we'll kind of reset it and then put a cast around it for several weeks until it grows back together. Now you have like a little bitty knot on there. We'll always be able to tell that you broke your bone in an x-ray, and everything's cool. But if you break it along these epiphyseal plates, your body doesn't replace it with kind of the more cartilaginous type of tissue. It replaces it with ossified bone. And that means that like once it's a closed growth plate, I'll quit using the air quotes, then now that bone can't get any longer. So if I break my right ankle, pretend this is my ankle, and now that growth plate doesn't grow anymore, but on my left leg that I didn't break, it's still growing, then my left leg is going to be growing at a higher rate than my right one is. And that's going to be a problem as I get taller. At my age, if I'm 42, I'm 44. At my age, at 44, if I break something on a growth plate, it doesn't matter because I'm not growing any taller. So, yeah. Okay, now for new stuff. This is the last of gigantism, and then we get into acromegaly. But in this video clip, it shows some actual footage of Robert Wadlow. That's not Robert Wadlow. That's some other dude. But here, you can see Robert Wadlow. I'm going to go behind the camera to see if I can zoom in on that a little bit. There he is at a desk. His hands are enormous. There he is walking through a door. And he gets in his special chair there. I have to figure out how to get the audio to work. No. Long story involving HDMI cables. And so eventually what killed him, it turns out, was your organs aren't really designed to function in a nearly 9-foot tall body. And so, hang on, let me get everything back the way it was. There we go. And so his circulation was suboptimal. And because of that, it turned out that I guess he had some braces, like leg braces, because, you know, he's a tall dude. And I guess one of those braces had like rubbed some sort of, you know, some abrasion or whatever on his leg, and it got infected and he died. But because he had poor circulation, he didn't really know that he had rubbed that. Sort of like when somebody's diabetic, when you have poor circulation, you might step on something, get like a little pin in your foot and not know it, and then it gets infected and you don't know until like the infection's gone too far. So essentially it was a kind of a, if you're a giant, you can just die from failure of organs. At some point, if you're an internal growth spurt, you die. And that's essentially what was happening. Was he couldn't keep up with his body. His circulation wasn't working properly because of that. This is acromegaly. So once your growth plates are closed, then now your bones can't grow taller, they grow out, they get thicker. So female hand, female with acromegaly hand. She has man hands, if you've ever seen Seinfeld. This is the same lady. I don't know the exact age, but as time goes on, like if I cover that up, that looks like a female. If I cover that up, that looks masculine. You'll notice the chin and everything else. So this is a lady with acromegaly. She was probably really tall too. She was probably releasing extra growth hormone from her pituitary gland all along. And then once growth plates closed, then now acromegaly. So the cause of gigantism and acromegaly is typically that you're releasing too much growth hormone. And in like Robert Wadlow's example, his problem was he had a tumor that was pushing on his pituitary gland in his brain. And because of that, his pituitary gland was always releasing growth hormone. So he's basically growing to death. Acromegaly. And then somebody that you're familiar with acromegaly, again, I think one of the classes saw this, Andre the Giant. He was in that movie, Princess Bride. There he is with Hulk Hogan and Donald Trump. Who'd have thought? All right, that video gets us through the stuff that's pretty much old news. Our next video, we'll start getting into oxytocin and aldosterone and ADH, antidiuretic hormone, insulin, glucagon, some things like that, and cortisol, the stress hormone. Oxytocin and ADH are interesting because those are the love hormones. And it turns out that based on genetics, you know, are you big V, big V, big V, little v, little v, little v. For ADH receptors, we can predict the likelihood that you have a happy marriage or kind of a stressful marriage. and the likelihood of whether or not you're going to stay married or get divorced based off of your genotype for receptors for a hormone, ADH. Who would have thought? So thanks.